Good morning, everyone, and welcome to the next of our 2023 CEBRA webinar series, or as we like to call them, the CEBRAnars. My name is Richard Kane. I'm Principal Director of the Innovation, Research and Education branch in Biosecurity Strategy and Reform Division. Before we begin today, I'd like to acknowledge the traditional owners of the many lands on which we meet today. I wish to acknowledge, for me, the Ngunnawal people as traditional custodians of the land we are meeting on, and recognise any other people or families with connection to the lands on the ACT and general region. I also wish to acknowledge and respect the continuing culture and the contribution they make to the life of this city and region. I want to extend that recognition to the traditional custodians of all of the lands on which our staff and participants are gathered here today, and indeed to all Aboriginal and Torres Strait Islander people attending today's CEBRAnar. Before we start, just a couple of housekeeping items. If you can just keep everything on mute, there will be a chance for questions at the end of Susie's excellent presentation. No pressure there, Susie. Also, best to just keep your cameras off and everything until the end. It just makes it easier. You should be able to see the PowerPoint up there ready to go. If you haven't used Teams before, we also do have the chat function. So as things pop up, you can just put in the questions and we'll throw them at the end of the session. Alternatively, if it does come up when we do have the sessions there for questions, just put the use the raise function and we can itemise and go through the questions as they pop up there. All right. So yeah, just make sure everything's on mute. I think it all is, and we've got everyone in here. Now, for those of you who don't know, the Centre of Excellence for Biosecurity Risk Analysis, or CEBRA, it's a long-standing biosecurity research initiative, plays a vital role in providing the Australian and New Zealand governments with expert biosecurity risk analysis and advice that helps inform a range of biosecurity risk management activities. Today, we have the pleasure of welcoming Associate Professor Susie Hester, who will discuss the findings on the benefits and costs of European Wasp management using the Biocontrol agent, and importantly, being able to value the social and environmental impacts. Susie is also the Deputy CEO and Chief Investigator at CEBRA and has been part of the research team since circa 2009. She's also an applied economist, having studied economics at my favourite uni, I have to say, Susie, because I went there, UWA, but has also worked almost exclusively on issues related to invasive species management since around 2002. Without further ado, I'm going to hand over to Susie, and if you all just want to maximise your screens, you should be able to see the presentation, and over to you, Susie. Oh, sorry, just flagging, it is being recorded. If anyone has any issues, let us know, but we're recording it so that you can watch it or play it back later on. Thanks, Susie.
Thanks, Rich. And hi, everyone from Anaiwan country in northern New South Wales, the land on which the University of New England sits and where I'm presenting from today, from Armidale. I acknowledge the Anaiwan people and I pay my respects to their elders past, present and future. My presentation today is about a CEBRA project that was completed several years ago now, in which we assessed the merits of biological control of European wasps. By we, I mean a whole wonderful project team, but in particular, I'd like to thank Oscar Cacho and Peter Tate for their work on that project. They were key to the outcomes and they're key to the work I'm presenting today. So at the time this project was devised, which was around 2018, the NBC, National Biosecurity Committee, were reviewing their approach to prioritising pests for management, and not just pests to be prioritised for eradication, but established pests that are widespread in the landscape, and so where a reduction in abundance would lead to large benefits to industry, the community and the environment. The European wasp was identified as one such pest, given that there had been an earlier attempt at biological control. We'll hear more about that earlier attempt shortly. Happily, we don't have European wasps here in Armidale yet, but I know that they're a particular problem in Canberra, as evidenced by this photo that I took in March 2020 around the time of one of our workshops. It was taken at Canberra's balloon festivals festival early in the morning, and the wasps were loving the maple syrup and annoying the people. So to our research problem, we were tasked with thinking about the management of an established pest using classical biological control. And as we know, when pests become widespread in the landscape, biological control becomes really one of the only options used to deal with them. When I talk about classical biological control, when I'm talking about biological control, I'm talking about classical biological control as opposed to inundative, for those of you who work in those areas. Classical biological control is all about getting a biological agent to establish and control the pest so that there are no ongoing costs of maintaining the pest population at low levels. So it's a very attractive prospect for governments. We've had many successful biological control programs in Australia, and some of these are illustrated in this slide. The various viruses to control rabbits, myxomatosis, calicivirus. We have a range of weevils and beetles to control Patterson's curse. I think we all know about the use of the Cactoblastis caterpillar to control prickly pear. And there's a rust that is used all over the country to control the weed bridal creeper. So when we're thinking about biological control, we need to think about the biological feasibility, the technical feasibility. Will the agent establish, and as such, biological feasibility is a precondition for success of classical biological control. And then we think about the economic feasibility. So if the program's not going to be technically feasible, there's no need to go ahead and look at the economics. So our question really was, under what conditions is introduction of a biological control agent for European wasp justified? As I mentioned, we created a terrific project team for CEBRA project 190804, some of whom are pictured in the slide. We had a team from ACPO, which is where this project landed. Con was the project lead, the departmental project leader, and we had James Woodman and others from ACPO helping. We had Oscar, an applied economist from here at UNE, who is exceptional at bioeconomic modelling. Peter Tate, an economist from Lincoln University in New Zealand, where European wasps are a particular problem. He specialises in non-market valuation. Ray and Greg from Ag Victoria were our biocontrol experts, our WASP experts, and they also had knowledge of the previous biocontrol attempt. We had Darren Kritikos from CSIRO assisting us with Climax, and we also had Jim Barishev, who runs Core Enviro Solutions, who was an on-ground WASP controller. And he came to our second workshop. We had two face-to-face workshops in the end, one in 2019 and one just before the border closed in 2020, which was handy for Pete to get back to New Zealand. Coincidentally, Jim was asked to control a European wasp killer nest near the Treasury. And so we all traipsed down there after the workshop and witnessed that, and that's where this photo is taken. Now to the pest in question, European wasp, Vespula germanica. It's native to Europe, North Africa and Western Asia, and it's a social insect, and its characteristics of eusociality allow it to succeed as an invasive species. It has overlapping generations. Reproduction is restricted to only a few individuals, queens. There's cooperative brood care in colonies. We can see part of a nest in that top left photo. That's the nest, part of the nest we controlled near the Treasury. We watched it being removed, and it was the cause of all the problems at the balloon festival. So the European wasp has been a successful invader in Canada, the USA, New Zealand, here in Australia, Chile and South Africa. It's been so successful because it's adaptable to a wide range of habitats and climates, and as a result has had huge negative impacts on beekeeping. So it attacks bees and beehives, robbing hives of honey, killing bees and larvae for protein, and spreading bee diseases. And the bottom photo shows a bee fending off an attack from a wasp. European wasps also affect horticulture by their effect on pollinators. They compete for resources and predation with pollinators. Importantly, they impact on outdoor social activities due to their aggressive foraging behaviour. Again, another photo from the balloon festival, that top right-hand corner, where wasps are enjoying leftover maple syrup on a plate. And they also, so they interrupt outdoor social activities, they affect biodiversity, and they also can sting and cause allergic reactions. In Australia, the first European wasp nests were discovered in 1959 in Tasmania, and then discovered in the remaining mainland states by the end of the 1970s. Early eradication attempts were attempted in the eastern states upon discovery of the wasp, but fairly quickly abandoned as the pest continued to become widespread. WA is an outlier here. It has had an ongoing eradication campaign for the wasp since 1984, and that continues. In the eastern states, biological control was attempted in the late 80s, leveraging off a biological control program in New Zealand, where a parasitic wasp, Spicophaga vesparum, was released in Victoria in the early 90s. Basically, this involved releasing cocoons of the parasitoid, but the agent appeared to perform poorly, and establishment wasn't ever confirmed, and very little has been written about this biological control attempt. My understanding is that the program was not well funded, and that there was also likely to have been a genetic bottleneck. The program in New Zealand with a similar pest was a little more successful than ours. Okay, to the biocontrol program, and the reason for a closer look at biocontrol of European wasp is because an agent had already been selected and tested for host specificity. This process can take a lot of time, and the thinking was that we could circumvent this and come up with a faster program. So the agent, Spicophaga, is pictured in the top photo, and the cocoons from which it emerges are shown in the bottom photo. When the agent is released, this involves placing boxes of cocoons near European wasp nests. The biocontrol agent leaves the boxes, enters the wasp nests, and eats the developing European wasp larvae. The biocontrol agent itself has quite a complicated lifestyle, which we capture in the model, but which I'm not going to go into detail about here. So if we considered using this agent again, we may not have to do any additional testing. There'd still be costs involved and reasonably lengthy timeframes, but these would be reduced if testing and screening could be avoided. We estimated that if no new testing was required, the importing, mass rearing and release of the agent would cost around $650,000 over 3.5 years. If retesting was required, costs would likely be a whole lot larger and the process would take longer. Regardless of whether testing is required, any biological control program would have additional costs, costs of long-term monitoring of sites and additional mass rearing and release costs. In the early years, there can be some additional agents released over the first couple of years. And these are calculated, the cost of these additional costs are calculated in the model depending on the release strategy. Okay, so the goal of this work was to develop a model that would allow us to understand the biocontrol program for wasps and indeed other established invasive insects. So this slide shows conceptually the decision analysis model that we developed and used to analyse the costs and benefits of an EWASP biological control program using Sphaerophyta vesparum. At the centre of the model, we have the population dynamics model. We've got the population dynamics of the wasp and the biocontrol agent and the interaction of the two. The European wasp population is modified by management, and in our case, we're interested in biological control. The population dynamics is also affected by habitat suitability. So as the wasp spreads into new areas, the probability that they will establish depends on the habitat suitability. We used Climax to look at habitat suitability, and we were lucky that CSIRO by Darren Kritikos had already modelled habitat suitability of EWASP using Climax. So that saved a significant amount of time. We used other sources of data from ABS and ABES, which I'll talk about shortly, and we considered both market and non-market damages. Market damage are those damages, in our case to pollination, beekeeping and horticulture, that are easily monetised because we can use price data, prices from the market. However, damages to nature conservation, public land and use and households are not readily monetised because those activities aren't necessarily traded in the marketplace. Our model was considering the damage that European wasps do to the natural environment, to outdoor social activities and to households. Because of the tight time frame in which to do this work, just a year, we couldn't do any primary data collection of these non-market values, so we relied on a technique called benefit transfer, where previously estimated values are transferred to our context. And Peter Tate undertook this work. Just noting that this was the first time that non-market damages of the European wasp had been estimated. New Zealand had done some work several years prior in which they had estimated some of the market values. Once we have the benefits and the costs estimated, we can then understand what management of the European wasp might look at in terms of benefit cost trajectories. In terms of data sources, as I mentioned, we're able to use the CSIRO research on habitat suitability, and the map in the top left shows what this looked like. Unsuitable areas for wasp are in green and suitable areas are in red and orange, becoming less suitable in yellow and light green. The unit of assessment here was the pixels on the Climax map. We also used the National Land Use Map from ABAS, 1 kilometre squared pixels, of land uses and commodities across Australia. We used ABS data both at the SA4 level for agricultural statistics and at the SA1 level for the census data, because we needed to know about human populations given our non-market valuation work. Next, I'm going to summarise the population dynamics component of the model. starting with European wasp itself. We needed to model European wasp growth over time during a season and over many seasons. Most work in this area has been done by others always using a Ricker equation. So we were able to use this and benefit from that other work. And we had some idea of parameter values to get started with. I should note that most of this other work had been done elsewhere, particularly in New Zealand. So we didn't have specific parameter values for European wasp in Australia. The graphic on the right shows a normal seasonal cycle for the European wasp, an annual cycle. Queens emerge in spring, around October, and start a new nest. The queen lays eggs and workers emerge after 4 to 6 weeks. The nest grows over spring and into summer as the queen continues to lay eggs and those eggs hatch, with lots of nest activity in late summer. So if you're in a European wasp infested area, you will know that there are high rates of wasp activity in February and March. New queens are produced in late summer and early autumn, and subsequently mate and hibernate over winter. In the native range, nests usually die off in winter, but this is not always the case in Australia. In some of the mild climates, they can continue to grow and become supersized. So key model parameters for our population dynamics model include the growth rate, which will be affected by the biocontrol agent, winter mortality, carrying capacity of the environment in which the nest sits, the dispersal rate. This is the probability that the queen, when she goes to look for a new nest, will settle on a suitable cell, pixel, and the detection probability parameter. So while there's no formal management of a program for European wasp in southeastern Australia, when nests are found by the public or in public areas, they are killed, and the value of this parameter will be a function of the density of wasp nests. We used New Zealand research and some known facts about the Australian invasion to assist us with parameter values. Specifically, to get our spread model running, we were able to use reports in the Atlas of Living Australia and a genetic algorithm to calibrate the model. So this slide shows the ALA reports on European wasp presence over the last 60 years, noting again that we're only interested in the eastern states reports. So to calibrate our model, we set an initial nest in Tasmania and ran the spread model hundreds of times using random numbers to give us different parameter values. And we used a genetic algorithm to come up with the 5 different parameter values. So basically we started with plausible values, let the genetic algorithm model run to come up with a probability map showing the proportion of times that the cells were infested in the simulations. And this graphic shows what that probability map looks like. So this probability map, this gives us parameter values, I should say, that reflect what has been observed in Australia over the past 60 years. Okay, that's all I'm going to say about the European wasp growth model. To the biological control agent growth model, again, we've got similar parameters to the European wasp growth model. But unfortunately, on the biocontrol side, we don't have the luxury of data from the Atlas of Living Australia. So we've got parameters for the growth rate, winter mortality of the agent and the dispersal rate. And in the case of the biocontrol agent, though, we have two parameters that determine biocontrol effectiveness. One determines how the population of European wasp is affected by the agent, and the other determines the winter mortality of the wasp nests caused by the adult parasitoid. We did have some information from New Zealand to get started with finding some plausible values, but not much. So the big question we're asking here is, will biocontrol be technically feasible? And for it to be feasible, we need to have the growth rate, R, of the agent being greater than 1. So what does this look like? For our two biocontrol effectiveness parameters, I'm showing the feasible area here. If the growth rate is not greater than 1, the population of the biocontrol agent will not grow, and so classical biocontrol won't be feasible. Again, the graphic shows where the feasible rate is for our two effectiveness parameters. On the left-hand side, we're looking at winter mortality. Values of winter mortality that will give us a growth rate of above 1 are in this area. If our winter mortality rate is actually falls in this area, then biological control isn't going to be feasible. So in our model, we used values in this feasible area. On the right-hand side, we've got a similar diagram for the growth rate of wasps. And again, the feasible area is going to be above a growth rate of 2. This knowledge, I guess, we don't know exactly where these parameter values fall, these effectiveness parameters fall, but with this knowledge, with the knowledge we've determined in this work, scientists can help us understand the likelihood that we can achieve these parameters. But again, if it's unlikely that we're going to be able to achieve effectiveness parameters in these feasible areas, then there's no need to do any economic analysis. It's time to, I guess, look for a different agent or a different combination of agents. Okay, so putting the model into motion, this is just a graphic of what one of the simulations, what it looked like when we set the model going. Of course, depending on the parameters, the natures of the cycles will change, and we've got 2 examples here. As the populations of wasps decrease because they're controlled by the agent, there's less nests for the biological control agent to establish in, and with less agents, then the wasps increase, as do the agents subsequently, and so on. So you can see the cycles that can emerge from our model. So for classical biological control to work, you need to achieve some sort of equilibrium with the pests, as shown here. Okay, so that was the population dynamics part of the model. We then combined the maps with the simulation to work out the damages. I'm going to skip over a lot of these details, and I'll talk later on about where these details can be found. But I do want to talk about the benefit transfer process briefly. Okay, so benefit transfer was used to understand the value of the damage to nature by wasps, to outdoor social activities, and to human health. The steps in a well-done benefit transfer are given on the right-hand side of the slide, and benefit transfer can be done really badly. The key is to find an existing primary study that matches the site of interest well. So we were lucky enough to be able to use a study by Rolfe and Windle from 2014 in which they measured willingness to pay by Brisbane residents for reductions in impacts of red imported fire ants. Adjusting the values from that candidate study, from that study, is a really important part of benefit transfer. So Peter Tate did this work for us and adjusted the study sites for inflation, for income, for the characteristics of the policy site, and for the distance of household from incursions. The adjusted values are given in the table, and these are used with the maps to create a damage matrix for each cell, showing the willingness to pay for reduction in non-market damages in each of these cells. So as the wasps spread over time, largely uncontrolled, we're able to understand damage. To the management side then, to understand the benefits and the costs, we focus on 4 plausible biological control scenarios related to growth. These were in the feasible area and with plausible, other plausible parameter values. We looked at, so those control scenarios were related to growth, mortality and effectiveness of the agent. And I've, in shorthand there, they're low or high growth and mortality with low and high effectiveness. So 4 different strategies. We looked at the base case where we compared no control to outcomes with biocontrol for each scenario. In the base case, we released over 337,000 cocoons of the biocontrol agent to infested sites in year 1, and each scenario was run for 1,000 iterations over a planning horizon of 50 years. We also looked at how the release could be managed or could be undertaken, and we did this by considering 2 biocontrol decision variables, and they were the extent of the release. So choosing the top percentile of infested cells to target, how many of those would be selected, and how many cocoons would be released per cell, so the intensity of the release. And information from those 2 decision variables would give us a number of cocoons required, so their costs and the number of sites needed. To some results. Okay, and these are, I guess, yeah, these are contained in this table. Without biocontrol, the mean value of damages from 50 years of wasp spread is $2.6 billion, with almost half of this attributed to the damage caused by wasps to outdoor activities, use of public areas, followed by damage to pollination and soft fruit. It's really important to note the contribution of these non-market damages to that final damage result, because I guess often these types of damages are not valued because they're difficult to value, but this example shows the importance of calculating these for some of these established pests. Of the 4 biological control examples, the largest reduction in damages is from the HH scenario, so it's high growth and mortality rates of the biocontrol with high effectiveness. The other thing to note about this table is that not only does biocontrol lead to a reduction in damages, market and non-market damages, it also reduces the number of nests that private citizens and public agencies will need to destroy. The damage caused by 50 years of European wasp spread without biocontrol are unevenly distributed amongst the states, as shown in this figure. This reflects differences in land use and area, population density, habitat suitability, and the age of the incursion. You'll notice New South Wales bears the largest burden of damage to public areas, and the New South Wales public spend the largest amount of money killing and destroying wasp nests. The benefits of biocontrol will be largest for those jurisdictions experiencing the largest share of damages, and the distribution of benefits is shown in that right-hand side. Okay, to determine whether supporting the program is justified based on benefit cost ratios, we need to compare the benefits with the costs of the program. For two of our scenarios, scenario LL and HH, this graphic shows the benefits of biocontrol, the blue dots, as blue dots with error bars, against the number of cocoons of the agent released, which is the red line. Now, the results are encouraging, although these results are predicated on the biocontrol agent succeeding at becoming established. And we should also note that we've, of course, missing error bars around the red line. So more research is required to understand establishment costs and establishment feasibility. In summary, our results suggest that provided conditions for technical feasibility are met, that we can get agents to establish, economic feasibility is high. There were some recommendations from the project. The first of these is that additional scientific research really needed to be undertaken to refine the growth rate of winter mortality of the biocontrol agent and its effectiveness at reducing wasp nest growth rates and winter wasp mortality. We also recommended that case studies be undertaken to gain additional insights into parameter values. And as a first step, we were suggesting analysing the ACT dataset that is collected via the eWasp reporting app. And that is something that Jim from our project team and his wife are in charge of and report on every year. And I would imagine that many of you in the audience are familiar with that app and maybe have used it. We also recommended that the department develop closer contact with New Zealand experts who are doing ongoing research into European wasp biocontrol, and this includes their research into other biological control agents, a mite, a nematode and a hoverfly, and those names are listed below. Outputs, well, there have been many outputs. Importantly, the code for WaspSim, the MATLAB model behind the population dynamics of the wasp and biocontrol agent, is freely available on GitHub, along with instructions for its use and for the use of different data sets with that model. We've put 3 journal articles out, one containing about the biological model, one explaining the use of WaspSim, and one which we hope will come out, it's currently under review, that looks at the economic feasibility of the biocontrol program. And I'm getting close to the end, Rich, some acknowledgements. The project team were wonderful. I'd like to acknowledge them again, but there are also various others who assisted us during this project. Christina Baryshef, who provided the ACT data to us, although we haven't done anything with it because we require some extra funding. I'd also like to note that David Cook from Deep Herd in WA was really useful in reviewing this project for us and discussing his experience with the WA eradication program. We also had Chris Baker assist as well and come along to our workshops. Okay, that's it. Thanks, everybody. I'll stop presenting.
Well done, Susie. Thank you very much for that. That was excellent and perfectly timed. Lots of claps there. Now, if everyone would like to, you can either just write in your questions as we go along or put your hand up. And normally I would go to the questions that are written, but I know we've got someone who's had their hand up. I'm just going to quickly double check here. I think it's Farzad. Would you like to have first crack?
Apologies, that was a mistake. Yeah.
No, ask a question while you got the chance. Right. I'll go to Nicholas Rush has just put in the chat then just while we're doing that to say fantastic talk, Susan. Just wondering what makes the tropical climate unsuitable for European wasp and how a changing climate is predicted to impact wasp distribution and how this may impact a biological control effort. Thank you. Thanks to Nicholas.
God. That sounds like too much information for me. I'm just quickly looking through some of my material. There might be others in the audience who understand that. I think it's probably related to the food available, and it's a temperature thing as well. I could dig it out and send the person who asked the question that information, but I'm sorry I can't be more helpful.
Nicholas, we might take that one on notice. Excellent question. Another one for you, Susie, is what measures would be in place to ensure that introduced biocontrol agents target European wasps specifically and do not harm native or beneficial species?
Yes, thanks. My understanding is that that's part of the rigorous selection and screening process, and that's why it takes so long. I'm not a biologist, so I don't fully understand that process, but I understand, given some of our past failures, that's a really time-consuming process. And I know we can shortcut that process a little bit with and leverage off the New Zealand program, but again, it's, you know, we need to rigorously test that. If there's anyone in the audience who would like to add to that, that's fine. I'm happy for that to happen.
I'm going to throw to Nigel while we wait for that. If anyone does have anything else they want to add, put it in the chat, might make it easier. But Nigel Ainsworth, I can see you've got your camera up, so fire away, mate.
Hi, Nigel.
Thanks. Hi. Yeah, you're right with the testing. I haven't been involved in testing of predatory and parasitoids, but I've been involved in plenty of testing of weed biocontrol agents, and they are indeed tested to death. But what I just wanted to point out is your comment at the beginning about classical biological control doesn't have ongoing costs. That's completely true when you're talking predators and parasitoids and herbivores. But if you're talking about things like calicivirus or rust fungi, it does have an ongoing cost because resistance arises in the target pest, and to maintain the effectiveness, you do have to keep often releasing further strains. But that's just another reason why this agent would be really good, because that wouldn't be an issue.
Thanks, Nigel. Yes, happy to be corrected on that. And the other, I mean, I was, I guess, new to the fact that there was another type of biological control, and that's inundative biological control, often used in agriculture, where you do keep adding, regularly adding to your agents. They're bred and put out there to control whatever you're controlling. So that was interesting.
Susie, just got another hand up. I think it's Dean Payne. So, mate, over to you.
Hi, Susie.
Hi,
Nice
Dean.
talk. It was good to hear from you. One of your table in the middle there, where you're talking about the total cost of status quo versus the reduction in cost based on the 4 scenarios. And I didn't do this in my brain. I'd like to convince you that otherwise, but I actually used a calculator. And I think the total cost was about 2.6 billion. And the best case had a reduction of $95 million. And so that was about a 3.5% benefit for the best case. And I was a bit surprised, that might be a simple analysis, but I was a bit surprised that there would be so little benefit. And I was just wondering if you could comment on that.
Yes, thanks, Dean. I guess we had selected our scenarios, our 4 scenarios based on technical feasibility, and that was the best outcome of those 4. As you would appreciate from the presentation, that this project dished up so much data. It was incredible. We didn't go back and find the best scenario that would give us the highest damage. So I would say potentially there are others that
so you're kind of conservative in your estimates. That's what you're saying.
Yes, yeah.
Okay, thanks.
Yeah, noted.
Right, thanks, Dean. Any other questions? Don't be shy. We had one other one around how does the behaviour of European wasps differ from other wasp species, and how does this impact management decisions?
So New Zealand has another wasp that is quite nasty as well. The name is escaping me. I'm looking through my notes. Vespula vulgaris. We don't have that here, but in New Zealand they are aiming for biocontrol that deals with both. So I think the impact of vulgaris, it can be just as bad if it establishes.
And just because I'm not even going to attempt to say that one, Susie, please, but that was another question that I had with a couple of people.
Sarcophaga. I don't know of any. No, I don't think so. No, we would have found them if there had been. The biocontrol agent, Sarcophaga vesparum, did persist in some release sites in New Zealand for a little while, and we learnt a bit about the agent that way. But the issue was, I think there was a genetic bottleneck, that the original agents were bred from just one European wasp, or one biological control agent. Again, I'm not a biologist, but that had implications for how successfully they bred and established. So the thinking is, if the biocontrol agent, the same biocontrol agent, could be collected from other places in Europe and bred from a larger range of agents, that the program could be more successful.
Very good. Note the time. We do have another hand up. I'm just going to throw, I think it's Martin. Yep, it is Martin Steinbaum. Over to you, Matt.
Thanks, Susie, for the presentation. I was just curious if the estimates of cost were based on public perception of RIFA control. I would have thought they'd be very high. So why is the model saying that benefiting Germanica is not worth it? Is that what I'm understanding correctly?
No, the results from the model suggest that if an agent establishes, if we can get an agent successfully established, then the benefits of biocontrol will hugely outweigh the costs. I don't know if I've answered your question properly. I don't know that I completely understood the question.
Yeah, sorry, it might have been a misunderstanding from what you were saying. So it would be worthwhile undergoing biological control of European wasp.
Yes.
Great.
If we know that the biocontrol agent will establish, so we need more research on the biology of that biocontrol agent. Unfortunately, we didn't have it. So our results are predicated on successful establishment of that agent, but this may not necessarily be the case.
Yeah, it's the same old story. A lot of these biocontrol agents
Yes,
have
but
lower.
I didn't have. I had a slide of concluding remarks that I decided not to use, and inspired. And one of the lines on there was taken from discussions with Oscar, who said, and it says, uncertainty is not an excuse for policy and action. So I guess I'm channeling Oscar there with that one, that you know, it would be worth it to do a bit more research as well.
It's the same old problem for all us entomologists. Not too many species and not enough known. Thanks very much.
Well, happy to talk more later, Martin. Thank you.
Thanks very much, Martin. I can't see any other hands up. I'm just throwing back to the chat. Susie, see if there's anyone else trying to catch me out with any questions. Yeah, we've got one here from Nigel Ainsworth. Thanks, Nigel. Is there an element of habituation whereby a perceived impact on people of a pest is high for a newly arrived pest, but declines over time as people become used to it and adjust their behaviour?
I imagine there is, but I don't live in a place where there's reefer or European wasp. So I guess people have to adapt and change their behaviour, but and maybe they adjust their outdoor activities to when wasps aren't present. In terms of input of fire ants, people don't have that luxury. I'm not aware of research that discusses that, Nigel, but I imagine there is an element of habituation. I'm not sure how that would play out in a benefit transfer or valuing non-market damages, but it's an interesting question, and I will pose it to Peter, the expert. Argentine ant, yes. And we just get used to it. And maybe, I don't know much about the Argentine ant, but it may be that it's not as bad as the reefer. Nigel also says, as a Tasmanian, Martin, as a Tasmanian, I have lived with Germanica all my life, including here in Victoria, and you never become complacent of them. Okay, thanks, Martin.
Excellent. And I think I can see a hand up. Sorry, I'm just getting jumping screens here, but I think David Toulon, if you want to.
David Toulon, yeah.
Yay, hi, David.
New Zealand. Hi, great talk. Thanks, Susie. Yeah, and I think most New Zealanders would say you never get used to wasps. I do have a question, though. It's the obvious one, and it's about the biosecurity, biodiversity impacts, because your value for that seemed quite low. And I mean, I was up in the mountains recently, and the number of wasps that were just destroying, that were just feeding off the invertebrate populations there was just phenomenal. I mean, unbelievable. And I mean, I know, of course, that's really very difficult to establish an economic impact value for that. But I don't know if you've got any comments on that. It just, you know, that impact to me would just about blow away everything else that you've got there by a long shot.
Thanks, David. Yes, I know New Zealand is different to Australia in some respects, and I think we're still learning about the effects of wasps on biodiversity. Recent research that came out after this project showed evidence of wasps on breeding on carcasses and disrupting natural other existing processes for carcass decay. European wasp nests survived the bushfires in 2019-2020. I suspect we're still learning. I know it's a bit of a cop-out, David, but again, some primary studies on data collection to understand those impacts on biodiversity would be warranted. Thank you.
Thanks, David, and thanks everyone for your questions. And I think just sort of closing, I can see one here from Martin, again saying that he's used metabar coding to quantify the diet of native insects here in Melbourne, and he will write up that shortly. No pressure, Martin, thank you.
Send it through, please.
Yeah, yeah. And look, for everyone who was asking before, it is recorded, will be available. I think Lisa said also the slides will be available, Susie, for people upon request. So we'll make sure we do get those out. But I do like to try and close on time if I can help it. I think we're spot on. So I'd just like to close by thanking Associate Professor Susie Hester here for an excellent presentation. Lots of questions and interesting ones at that in terms of value there. And it was really good to have everyone here today from quite a diverse background. The recording, of course, will be sent out shortly by Lisa. If you've got any other questions, let us know, or anything else you can put in the chat. Details on our next CEBRAnar will be coming out in September. We'll try and time it around the school holidays. We'll see how we go. And we'll make sure we send that out to everyone. But just thank you very much, Susie. Excellent presentation. I hope everyone enjoys their Thursday. You can all go off to lunch and have a coffee as well. Thank you.
Thanks for listening, everyone.